which are taking place in Pioneer Canyon in Greater Farallones National Marine Sanctuary, and then several dives in Monterey National Marine Sanctuary, including Davidson Seamount and the Whale Fall. We will be joining through the science chat as we have been on this one for those two, and then we'll be back on for the Channel Islands.
Excellent.
So it's going to be a busy fall.
Yeah.
It's actually great to be able to, even with the weird circumstances we find ourselves in, to participate this way from the comfort of one's living room.
Yeah, it is a very strange year for field work all around.
Yep.
Mm-hmm.
Head back down again.
Yep. This is a pretty sharp.
You seem to have found super terrain.
Be offline for a minute. I've got to change microphones here.
Okay. That is one of the challenges of broadcasting from home, is getting your own equipment to cooperate.
Yeah.
And I find it doesn't always cooperate with every system you have set up. For example, my system cannot deal with Google Hangouts, but it deals with everything else just fine, which is always a challenge when certain employees of the government can't use anything but Google Hangouts. And so
yeah,
it's almost having to have an entire second set of equipment just to work with one software platform. But worse things have happened.
OET is very much Google-centric. All our stuff. Rocks. Meets.
Google me.
yeah.
I have a Google phone.
Yeah.
Me too.
It just took a picture of the slope map for me on my Google phone.
He pays for it.
Yeah, I pay for it. I see so many tinamores here. In like gelatinous Texas.
Dragging the rear in the mud. I don't think we're going to see a whole lot this stretch here.
Yeah, this little kind of dip looks pretty steep. But I think as we get on the other side of it, this next spine looks less steep.
5 meters on the back of us all of a sudden. Going down.
Okay.
I mean, if you wanted to follow along this contour to the next spine and then come down on the spine, I'd be fine with that too. If you want more vision.
Honestly, be better looking out the rear.
Yeah, I mean.
I can see the slope kind of coming to the right of me right there, so I don't want to go down too much more.
Yeah, that's the other thing.
It's not uniform.
I'll have to
Just
wait.
Yep. See it on the MESA.
Ferguson's almost in the dirt.
I mean,
Yeah.
Okay.
Does it level off a little bit? You can't really follow the slope here because Ferguson will be in the dirt.
Yeah.
so if, yeah, I mean, my suggestion just is if we wanted to go kind of 060 and keep kind of tight to the same contour on the ridge, and then kind of descend when it's more gradual on that spine. But it's my only suggestion. Let me. See, like.
Yep, bouncing around.
Yeah.
Yeah, that's weird. It's like,
Go by this.
okay.
Bouncing off the side.
All right.
So are we headed for 3.05?
Yeah, we're heading for 3.05.
Okay.
So we, yeah, we're kind of heading there efficiently in a crow's flight.
Yeah.
But yeah, if you wanted to keep on this kind of contour and then descend down on that next spine, I have this slope map loaded. You can kind of see, be a little ahead of that target.
Is that?
So kind
Yeah, I my only concern with doing that is that looked like from earlier the sponges were in the very,
okay.
very steep terrain, so we don't want to miss that entirely.
All right. Well, it does ease up a little here, so
Yeah.
hopefully we'll get a nice view of the bottom soon.
Yeah.
So the sponges are on these deep slope areas to kind of facilitate their, you know, filter feeding.
Yeah, it was the wall seemed to stay a little cleaner, so it's not buried in mud, and they require hard bottom habitat. And then they probably are getting some sort of change in current flow by being on the wall that makes it more efficient for them to feed.
Right.
You'll often find corals placed strategically the same way, where they tend to grow well in areas where the flow rate is kind of optimal for their feeding.
Oh, cool. So many jellies.
Mm-hmm.
And a chimera just to say hi.
Oh, yeah.
Oh, yep. Look all photogenic up in the water column. So I will wait for Liz to get back on with her microphone, but we might consider the option, since all we're seeing blue water, of coming out off the wall to drop down and then coming back in at it.
Yeah, I think that would be a smart idea. But
yeah,
because we're essentially doing that, we're just
yep,
not.
Yeah. So I was thinking about that because we're not seeing the slope at all here, so it may
You
be
can see
more
it in the
efficient.
rear cam,
Yeah.
Look in the rear cam, you can see. There was a sponge
She
back
just chimed in and said yes. So, so why don't we go ahead and set up to do that, to just come away from the wall and just do the drop off the wall and then come back in at it, if that will work for you.
So this current wall, so I could bring us down to that old target, that 3.04 would look like the base of that. Does that look good to you? And then
we can see,
Yep,
you can see animals in the cameras. It's just you can't see them forward-facing camera.
Oh, there's huge sponges.
Yeah.
See in these other views,
Oh.
you look out the rear and look at the down camera. You just can't see them out the front.
Mm-hmm.
Yeah.
You can see in the rear camera the tether's like right in the mud. Like I can't go any lower.
Yeah. so I'm going to look at the chat here. Liz, do you think we should just start at 3.04 then, and then move up to 3.05, just ladder? Then we'll be laterally along a contour and not out in space.
There's another one there.
Yep, she sounds good. So let's we can go ahead and come kind of off the wall and drop straight down to 3.04, and then we'll just pick up that wall and move to 3.05 from there.
Okay.
Just gonna hold that.
Yep.
It'll be
It's
more
gonna be
comfortable flying.
140. Should I just track a line for that?
Yeah.
So for those watching at home, if you are on the Nautilus Live website, you'll see a picture of the Hercules down at the bottom of the page. And Hercules is quite a large vehicle. So while the front, which is the camera view that I'm looking at here, looks like it's way out in the water column, because it kind of is. The back of the vehicle is just above the seafloor and very close to the wall. So we are adjusting strategy to get repositioned in such a way that we can face into the wall again and move more easily. And that will allow us to start doing our surveys once again and hopefully collect some more sponges.
You can see some in the Argus view, just barely there.
Hopefully there's still sponges when we get to the bottom of this feature.
I'm
Mhm.
sure we will. It seems
And
like
at least we have the camera view from the back to know what was up there.
We don't now. We're just
Well,
going.
They screen grabbed it.
Going at a fixed depth.
Yeah.
Just gonna go out and go down.
Yeah.
from
I'm just saying we previously.
from the first transect in this area and from the camera views that we sort of have, there are a lot of these really big white structural sponges. So I have good hopes.
Well, you
Yeah.
see some in the Argus view.
Yeah, exactly.
Yeah, I just pulled up Argus and I just saw one go off the end of off the bottom of the screen there.
What did you call it?
Me?
Yeah.
Oh, I just said
the species name. I didn't.
Oh, I was just
Oh.
I don't
Okay, how far are we
I
gonna
can't give
go?
you the species name of
Okay,
a sponge.
so
I'm
we're
very
gonna
sorry.
jump down to here. It's gonna be like 80 meters
Okay.
only.
So are you going faster now?
He's going 0.3 now. And then I was just gonna bring us like that. Unless they want to go up a little, and then, but we don't have much. Wow. Siphonophores, ctenophores, sedipitenophores.
Anyway, I was just describing the
see a-
sponges as
Yeah.
structural.
Oh, structural, yeah.
Yeah.
They definitely have structure.
Yeah.
It's like manifolds and.
Is that a different bearing? What's this bearing?
This is 140. Oh, did you not get that? Sorry.
I think that sponge that we saw, that was the big white one, looked like one we would be calling heterocony, although from Argus above, it's a little hard to tell.
Yeah, fair enough.
Especially with all the critters in the water.
Meredith,
Just had a
can
little
you hear
eel
me?
pout float by. I can hear you, Liz.
Ah, yay. I'm back on, I
Yeah.
hope, for a while.
Excellent.
And be sure to tell me if I'm fading in and out. I can try yet another set of headphones. I've gone through 3 here in the last 3 days, thanks to the miracle of Amazon. Delivered at 10 a.m. to my doorstep.
Seems like this one's all right. It's a little crackly, but you're understandable for sure.
on my last watch, I tried using my fancy lab headphones that I'd retrieved from the lab. They don't have a built-in microphone, and the computer decided that there was one, so no one could hear me because I didn't have an active microphone.
Oh
So
my.
I switched back to my little cell phone headphones, and now it's working fine. Do you like all the little critters we have floating by? It's been largely these ctenophores, the marine snow, but we had an eel pout a few minutes ago, hanging in the water column. And we've gotten an occasional hake.
Yesterday on the dive down to the 1,000-meter site, we had quite a few of the giant appendicularian or larvacean housing that I'd only read about and never seen on a dive, which is really exciting.
Seeing some Keating nests too.
Oh, nice.
Little arrow worm.
Just
one was just out of view.
Yeah, lots of little siphonophores and salps.
Yeah,
and
it's a
like
huge
that around
diversity.
too. Yeah. Earlier there was just a mysid swarm around Argus.
Oh yeah?
Yeah.
Definitely productive environment.
Mm-hmm.
I got stuck in a mysid swarm scuba diving in Port Orford to do training dives. And there's so many mysids, you couldn't actually read your own gauges, so we kept on having to abort all the dives. It was even worse because there were whales swimming around feeding on us, and if we couldn't see our gauges, the whale couldn't see us. So we figured we were training for something we didn't want to do.
Yeah.
Oh, that's a good story. Caught in a swarm.
Done a little ROV work where we got the ROV into a swarm of mysids, but then we had copper rockfish that just came in and were all over feeding.
I'll bet that was spectacular to see.
Yeah.
Oh yeah, it was fun to watch.
Where there's a swarm, there's predators.
Yep.
Mm-hmm.
Although the YouTube video of a whale eating a small ROV would definitely get some hits. It would not pay for that new ROV.
Yeah, and if you guys want to, I think we can settle down soon. If you don't, I don't know about that 3.04, if it's at all useful to get down there, but we're at enough depth that we're kind of safe to travel back to that 3.05.
Yeah, just look back over. Oh, yeah. I think we could just, settle and lateral straight over there since we knew we were seeing sponges up above.
Okay. I'm just going to change our bearing and not hold. Or do you need me to hold? We're going to go back to the bottom and then just move over to 3.05. What? No, this is on a, I mean, it's the same contour. It's a little wavy, but should be helpful. So I'm going to keep the line and just add a, it's going to be
Go down.
Okay.
100.
Yeah, Kurt is pointing out online that the waypoint itself is less important than just following and looking at the steep terrain. So this sounds great.
Yeah, I assume so. I just wanted to confirm that we can get back on the bottom.
It's also easy to have something concrete.
The steeper, the better.
Yeah. I
And
think
the deeper, the
maybe
better.
after last night's festivities, we're being cautious.
Yeah.
But it's good to do things right.
Hey, Meredith, I've changed microphones again. Can you hear me?
Oh,
You sound good.
I can hear you fine.
Thank you.
I'm great.
Travel into the snowstorm.
One of the more entertaining.
Seeing a lot of those.
Go ahead, Liz.
Yeah, it's very restful, isn't it? We're seeing a lot of these low bait ctenophores. Not so many of the sea gooseberries, which are, you know, Pleurobrachia bachii. But they're beautiful.
Yeah.
Oh, did you?
I wonder if they're all clonal, these low bait ones. It's hard because some of the, there's this one order that's called Thalassocalisi or something, and there's one species, but it looks kind of like a low bait ctenophore. And I'm always questioning because I have seen them on the Pacific and video data, and always just I can't really diagnose between those two. But anyways, yeah, it's a lot of these low baits. And
I may have just seen a pteropod go by.
Yeah. I love the Physonex siphonophores because they just like have these, some are totally vertically oriented and some are more like sinewy. I wonder what's, you see like there, they're all vertical. Picking up a lot of stuff, probably. Foraging.
Yeah, it sure looks like a time of plenty right now.
I'm hungry. No.
Well, it's why we get an oxygen minimum zone along a lot of the West Coast, is because there's so much production that then sucks up a lot of oxygen as it decays. That it's really unusual on this coast. Well, it's typical of a place where you have a high productivity, like upwelling, but it certainly changes the dynamics across the West Coast. I was reading that paper, Andrew, of yours and Lisa's, on oxygen minimum zones on the West Coast.
And they're fascinating environments. One of the most striking images I've seen was a picture from a plane of upwelling off Namibia. The upwelling was so strong and the oxygen minimum was so strong that you could actually see sulfur oxidizing bacteria on the surface of the ocean.
Wow.
Oh, wow. I did my dissertation on hypoxia in the Gulf of Mexico.
Let's go.
Cool.
Yeah, the
still sort of downslope a bit.
We should be.
Oh, I'm seeing the bottom.
Yeah. Should be coming back up slope as we.
Seeing some sponges already.
Up and down and up and down.
Yep. One right ahead of us.
Perfect.
It's like a ghost for
Excellent.
Halloween.
It's true.
Nice big header coney. Some Sphaeria. Sphaeria doesn't look very healthy.
So, one of the things I'd like to do, and I don't know if we can do it, is I'm interested in kind of quantifying what's dead, what's alive in this sponge community. And if somehow we could do a transect across one of these sponge beds, it would be peachy, because I'm trying to kind of decide or get a quantification of who's dying, who's living, and who's young.
So would a transect that's
I know it's
along this slope, and would that be useful?
That would be perfect.
Useful. Okay.
Yes. So I just thought I better tell you my goal here.
Yeah. We want to meet your goals.
Because if we see one of the important sponges or corals, we can stop the transect because I'm sure collections are more important. But you can see we're seeing some dead sponges,
Yeah.
some live sponges, some young sponges. I'm trying to get a handle on that, what that community of sponges is, what's happening.
Yeah.
So just tell me, Meredith, if you want to start a transect and when everybody wants to, and that's good.
Let's say we're just moving laterally across the slope, and they'll be a little up and down, but shouldn't be too bad. We can just go ahead and start one right now, and we'll pause if we need to do any collections. But why don't we just go ahead and drop a marker and start a transect?
Okay.
And we'll try to make it 200 meters.
Yeah, it's going to be 110.
Yep, just what we're doing would be great.
Yeah. And so transect number 3.
Yep.
Oh, look at all those. You can see a bunch of sponges in the park.
Oh yeah,
Yeah.
it's like a sponge field.
Nice.
Yeah, we don't call this a sponge reef, even though it's really dense, like a lot of the sponge reefs are, because it's not really built up decks of old sponges, we think. But that's why Andrew was thinking, I wonder if we could take a core here. We have some sub-bottom information that says this is probably not a sponge reef, and I sense from looking at this, getting a core in it, we've tried before, is like zero possibility.
Looks
But
like we're kind of
they
missing
just-
where all the sponges are by meters.
Yeah, if
Are they
just
above us
higher
or
than
below us?
if
I
you'd
think
like
they're
us,
above us.
so if
See
the
on Argus screen.
3.05 is not a very important, specific location, I can just bring
No.
us slightly upslope and change our bearing so that we're kind of going more like 050 on this, like this line I'm drawing, so that you can get
Yes.
more of a vertical gradient. Okay, let me do that.
Balls right
That
there.
would be
Yeah.
great.
Yeah.
Yeah,
Awesome.
see, he's right up against the wall.
Yeah.
Well, that's
Might
good.
have to come up a little bit.
Oh,
I don't want to do that.
oh yeah, Argus is in between you and the wall right now.
So that would be better to go more up because it seems like I'm kind of going down slope right here a little bit.
So
Going across, but the train's sort of going down.
yeah, so if I did 040,
Yeah.
is that good?
3. 0, 0, 3, 0.
Yeah, let's do that.
Should we restart the transect, Meredith? Or do you think that's okay?
I think that's okay.
Yeah.
Southwest is really good about pausing and just connecting little segments if they need to.
Yeah. Okay, great. That's my sense. We've done some pretty bad transects before, but we've managed to stitch them together.
And when you're working on steep walls,
That's a
like
problem.
this,
Yeah. We never do a steep wall like this with the AUV, but one thing we don't have is we got no problem driving a straight line as long as it's not a wall. So and nobody gets to stop for collections with an AUV, which makes the line very straight.
You know?
To know that we may go and do the same thing. I think it's
We
good.
can only do what we can.
Yeah.
Coming up.
And that's a sharp chin, is it, Erica? Do I see the V behind the eye? think I do.
There's a
That's
little
one
red
of the
swifty there.
heteroconate. Oh, be sure to tell me if you want to stop. Or tell them, not me.
Yeah, that's the species that we actually have quite a few of from the trawl survey because they're just so common. That's interesting rock texture there.
Yeah.
All of the holes.
These are so cool looking.
Yeah, this is very. Should I?
You're gonna have to slow down.
Yeah.
Can't do this at any sort of speed.
Slower is fine. It's just amazing to me that these guys can cling to the wall and hold their structure like this.
Why don't you go back to step position?
It's like they're dripping off the wall. They're really standing on these big vertical trumpets.
Seems like
Before the
wall's kinda
samples,
topping
we
Sorry,
off.
what?
Seems like the wall's kind of topping off right there.
Yeah. See
So instead of moving up, if they were topping out the wall, instead of moving up back onto that kind of flat slope, maybe we could lateral along it up here.
Yeah, it seems
Sounds
like
good
this
to
is
me.
the top. This seems to be the top, at least locally.
yeah, and then we could lateral here. It's fine.
There's a little swiftia just to the right of the lasers. A little red fluffy thing is a small coral.
So Abby's on the chat now. She's our sponge expert.
Whoa, look at that. So cool.
Isn't that gorgeous?
They're almost
There are
puppet-like in the
places
stuff.
that where you'll see 4 or 5 rockfish stuffed in a sponge, but much in these open heterocone, more in, or heterocone.
I don't know.
I'm told I pronounce it wrong. But he looks very comfortable bedding down for the night. But in some places we see 4 or 5 rockfish stuffed in, nose down. You can just see their tails.
Okay, well, so I'm gonna have to come up over the top of this just to get back at the targets here. You're gonna have to. What do you want to do?
Is that a sablefish?
Either get
Yep, that's
targets
the
down to
sablefish.
me, or I'm gonna have to go over the top.
Bridge nav. Can I have a ship move 20 meters just due south? Great, thanks.
You want to take some time and look at this because we want to get the position better here between Argus and Herc. So we have a little time.
Bob, you're still on an SPL if you would like to be. Liz and Meredith, did you want to spend some time to look at this? We're waiting on Argus to catch up, so we have some time.
Did you say you couldn't hear me?
I couldn't hear you on SPL. Was I the only one?
Yeah, I
I'm on
couldn't hear him
SBL.
either.
We hear you now, Bob. You just had to get your mic closed.
It's a what?
Yeah.
You want to zoom in, Dave? We're going to have to wait for
I'd love
the ship to get settled here.
Yeah, if you could zoom in, that'll
Okay.
be lovely.
Okay.
It's such a beautiful picture.
Yeah, it really is. The texture's amazing.
Be great if he'd turn around for his close-up.
Can we turn off the lasers for a moment?
Yep.
Thank you.
Erica's saying that that is either an aurora or a split nose.
If he'd turn around, we could tell.
Mm-hmm. I like that these two sponges almost they're growing together.
So we've seen a couple of these that have slightly different color morphs. Are those, is that indicated by age, or can you learn something when you have two right next to each other that are one slightly yellower than one that's more white?
I don't know. I'll let Abby answer in the chat, but we haven't figured that out quite yet, whether the color morphs are morphs or different species. I know the flatter ones that are more yellow are very different, but I don't know about the color in these heterochrome. We'll see if Abby weighs in. What's interesting, these, you know what these look like to me? You know, the styrofoam mattresses that are kind of have little
Oh.
processes on them.
Yeah.
It looks like this guy is sitting on his foam mattress.
Yeah.
From a rockfish perspective, it probably is like a comfortable mattress.
Yep.
Yeah, he's not trying to
Okay,
stay
you have to
stuck on
zoom
that
out,
wall.
Yeah.
Gotta-
Ooh, an egg crate. That's what Kurt said it looked like. Oh, that little yellow sponge. Can you zoom in? If you just zoom straight now, there's a tiny bit of yellow on the left-hand side of the biggest sponge there.
Okay.
And it's kind of bright yellow and it's tiny.
Yeah.
Yeah.
It's above the threesome.
We can turn the lasers back on, by the way.
Back up.
It just looks so bright yellow
Okay,
to me
Dave,
there
zoom
for a
in.
moment. Yeah.
Yeah. Just peeking out there.
Wow.
All right, I'm at the back off. Zoom out.
Thank you.
Did you see it?
Yes, I did.
Okay.
Perfect.
Okay, so we're a little better position now.
Yeah, we are. You know, it's just, I don't know if you just want to follow along visually, because it's going to be kind of this.
Yeah, I don't think we have accurate enough
Yeah,
maps there to
exactly.
like follow
Well,
along
and,
ridge
I mean,
crests
even
or
on the
anything.
map, it's wavy here.
Yeah.
So, you know, if we can move along to the next, you know, objective target.
Okay.
And
what's the general bearing?
all right, so it's going to be like 085, almost due east, and
Okay.
but I'm thinking I'll do something a little more like 075 to keep us along this.
Yeah. Okay.
Is that all right?
Yep.
With her? Okay.
Yeah, Abby says in the chat that the Africalistes are white, so the yellow is helpful to distinguish the heterocone. Although shallow, I've definitely seen yellow Africalistes.
So have I.
Of course, it's blue water out that way.
Well, I'm trying to hopefully follow this.
Yeah, I'm just going by the sonar.
Yeah.
Sonar says it's going out the blue water out that way.
All right, let's bring this in a little bit more.
So Kurt gave us a heads up. He just said, and FYI, the targets were selected with bathy, not as good as what you have now. They are only a guide. Safest method of travel is good. I'm sorry, I interrupted you, Meredith.
I was just pointing out we have all these Sola sea cucumbers mixed in amongst our sponges.
Yeah. Kind of
And the
purpley
feeding
white.
tentacles.
And a lot of crinoids living on the sponges.
Yeah.
Mm-hmm.
I would like to poke the crinoids occasionally and make them. They don't look like they should be able to swim away, but they do, and they're pretty. They're kind of beautiful and balletic as they move away.
Mm-hmm. Swimming with their arms.
Yeah.
Yes. I'm going to say that's a rose thorn, that little fish with the white spots. But if I was careful, I'd just call it a sebastolobus, because little red rockfish with white spots, there are many.
The little brown birds of the deep sea, of the sea.
Yes.
Very much so.
But I have an ornithologist friend who says there's dicky birds, seabirds, and eaten birds.
Straightforward
He's a
taxonomy.
seabird guy. Yes. So the sponges that are covered in sediment are, I would say, most assuredly dead. But
Yes.
it's hard to know why they're dead and why some are alive. Is it just part of their natural life cycle? What's really going on? That one looks flopped over. Maybe it just outgrew his foothold. But you see, that one is still standing straight up as if it were alive, but it is dead. It is an ex-sponge.
So interesting that these sponges that are the species that can form the reefs when they are alive can be very, very friable, very delicate. If you were to brush by, they kind of fragment. But when they die, their skeletons fused and they become sponge-shaped rocks.
Wow.
It's coming up at us on that side again.
Abby, is
Lots of
that
nooks and
a
crannies.
rhabdo you want to?
So
Can we zoom in on those sponge barrels that are on that little rocky prominent there? And they look like hairy barrels. And they're just in the middle lower part of the screen right now.
yeah, we shall try.
And
Okay,
is that,
Dave, wanna
Abby, is
zoom
that
in?
an X?
Zoom in, Dave.
You know, that's not what I thought it was. I think that is
No.
just a sediment covered afro, not a hairy boot.
Hairy boot.
Oh, hairy boot. Okay, zoom out.
That's it. Thank you. Well, we're looking for something that looks a little hairy like that, only it looks like a little barrel.
Gotta catch up here. Are we still on the move? What's happening?
Yeah, we've done this for 30 meters now.
How big was that move?
I did 15, and we have 5 meters left and another 15.
Yeah, we got
Abby says
smaller
that many
moves.
sponges.
All right.
We're gonna stop in smaller roses
Yep.
here.
No, it's good to know we're going to do that.
Well, there's a
Abby
nice
said
big
many
piece
sponges
down.
are hermaphrodites. So when I said he or she, it's fine.
I once got into a thing where I wrote guys on a blog about hermaphroditic, organisms, and I was told it was pernicious of me to call it guys, and I've always been careful of that. I just thought pernicious was a very strong word. Not like I intended to
Yes.
be gendered about it.
Yeah. Well, anybody who's using pernicious in a blog post, I worry about anyway.
It's like, first I'd Google
comment, right? Yeah, exactly.
So that lacy looking sponge right up near the top is, I think, Faria.
Oh yeah.
Oh, I got my pronunciation corrected, I think. Say it again. Oop.
You want to zoom in, Dave?
And we've collected some of those.
Yeah, we have.
They have almost a kelp-like texture to them when we tried to collect them. It makes me think of the sea lettuce you find in the intertidal. Only they're white.
They produce
And
a
it looks like
surprising
he's got
amount of
some.
mucus.
Oh, do they? And you figured this out because we collected them and what you got was a small piece and mucus?
Yep. They're quite slimy for being something that's so like delicate and fragile. Nothing like the mucus bombs we collected, but they pulled them up.
I did a lot of reading recently on hagfish mucus.
Ah, yeah.
Isn't it like the most per protein or whatever they put in? Like they're incredibly efficient at making things gelatinousy and stuff.
Yes, and according to Google, people they have in the mucus, they have they have skeins of kind of stronger material in the mucus, and they're trying to replicate that for things like
Tuxedo.
You got another move in?
Yeah.
sort of armored vests and things like that.
Oh, yeah.
But I've been reminded that there also, for hagfish, there's a commercial fishery for making eel skin wallets. And there was a famous road closure in Oregon when a truckload of hagfish fell over.
That.
And yes, and caused quite a tie-up in the traffic.
My mouse is not working.
Well, everyone, I think I'm going to sign off for the night. It's been really great chatting with you and an enjoyable wall of sponges. Thanks for the seep intermission as well. Good luck with the rest of the dive.
Thanks, Andrew.
Thank you, Andrew. This is, I guess, the last watch of this cruise. So I just want to congratulate you and everyone on doing just this. Was so well done, and your seep work was fascinating to me. And we should talk about some AUV data I have that may show some seep distributions. So we'll talk.
Oh, I'd love to talk with you about that. That sounds wonderful. And this has been really fun. Thanks to everybody at shore as well. Really making it, and it's been just absolutely spectacular to work with the Olympic Coast National Marine Sanctuary and everybody on it. So thank you as well.
Sleep well.
Where's all the good stuff?
Now the sponges are showing their
I'd have to fall off of here.
What's
I mean,
that
I'm weighing
yellow? Do you see up there?
Yeah.
Yeah.
Can we zoom in on that yellow sponge that is at the top of the screen now, the middle at the top?
Wanna zoom in, Dave?
So I am.
Abby, any interest in that? Let's see if Abby's still on. Yep, Abby's still on.
I'm having a technical issue right now where I can't move my mouse.
Oh.
So I can't do the screen grabs.
Oh, so maybe break this. So below your monitor, maybe try to break it with that plug out button, then try to grab it again, maybe.
Okay.
Oh, no, that is a yellow Michaeli, and she wants to wait for a yellow Starocalyptus, which would be a little bit more barrely looking, I think, and a little bit softer walls. Floppier, slightly walls would be what I think we're looking for. But Abby, correct me if I'm wrong. So thank you for the Zoom, and we've seen what we need to see.
Okay. Moving on.
Bob, I'm going to take bubble quick to check.
Yep.
I'd be giving us some sort of view on what the best areas for feeding for some of these are, because we came right around that corner and then they all disappeared. or they're underneath us, one of the two.
Yeah.
Yeah, I don't
And
see
the
any in the
question
Argus
is,
view.
Oh, what's that in the upper right?
yeah. Oh, that's weird. Do you see that,
Take a look at
Abby?
that.
Yeah.
Can we zoom on that odd white kind of pipe structure?
Okay, zoom in, Dave.
I'm thinking we might. Wait a minute. I'm checking with Abby. Yes, a weird polymastia, she thinks. If we could collect a piece of
Oh,
this.
Okay, zoom out, Dave. How are we gonna collect it?
Well, I am.
I was wondering if it might be
I'm thinking this is a grab. Say
Yeah.
again, Meredith?
Oh, Abby is saying that she thinks it might be, it could be a fragment from a bigger sponge, so maybe try touching it and see if it's attached.
Okay.
So give it a whack if we could. Are you okay holding here, pilots?
Yep. Turned out this was a good spot. We're stopped, right?
Abby,
Yeah, we're stopped.
Abby says,
Okay.
give it a scientific
You want to
poke.
zoom in, Dave?
I think on NA86 they had something they called a wham stick that they
Yeah, it
would
doesn't seem like it's
pull out.
wasn't attached to anything. It's sort of
Loader.
Yeah, that's just for,
Yep,
for loading.
she's right.
Yep.
Yep. Okay. I don't think we need it then. Do we? Wait a minute. Let me ask. I'm sorry, there's such a delay. Oh, she does want a piece of it.
she wants a piece of
Or
it. It's pretty loose, so
well, you could pick the whole darn thing up.
okay.
She says it's already
Because
detached.
it's
so
an easy sample.
Data, do you know if this is sample 134?
This should be sample 142.
142. Got it.
Oops.
And we've got a lot of the bio box slots open in the starboard bio box.
Yeah.
But if it's kind of tenuous, then
Yeah,
we could also put it in the forward.
That would be better.
We could put it in the forward left-hand box.
So the problem with the starboard box is the vertical thruster on the starboard side.
Yeah.
It'll blow down on it. I can turn that off, but then I'm going to lose position on the wall, and then it'll be a big mess.
Yeah, then you can put it in the left-hand side of the forward bio box, bio box A.
Jake, you want to push it out? You give me a porch light too?